Welcome, students, to the last part of chapter 11, which is mainly really about how neurons kind of work together, how they talk to each other, how they cooperate with each other. So that's where we're going to kind of talk about synapses and postsynaptic potentials and neurotransmitters and neuronal pools and all that kind of happy jazz. A synapse is a connection between typically one neuron and another neuron, or one neuron and some sort of effector cell. We've actually seen synapses before when we looked at the neuromuscular junction. That was a connection between a neuron and an effector cell, in this instance a muscle. So that synapse connection between a motor neuron and a muscle cell was a type of chemical synapse. But in the nervous system, not only can you have chemical synapses, you can also have electrical synapses. And neurons can connect to each other in lots of different ways. They don't always kind of connect one end to one end. What they can do is connect kind of the end to the cell body of a neuron, the end of a neuron to the axon of another neuron, or the end of a neuron and the dendrites of a neuron. So really we have three types of synaptic connections. The most common type of synapse that we find in the nervous system is an axodendritic synapse, where it's the axon terminals of one neuron connecting to the dendrites of the next neuron. But we do also have axosomatic synapses, where the axon terminals of one neuron connect to the cell body of another neuron, and we do have axoaxonal synapses, where the axon terminals connect directly to the axon hillock and the axon of kind of the next neuron. So there's a little bit of terminology associated with synapses, and we use two terms to describe kind of where we are in relation to the synapse itself, to that kind of cleft there that's full of fluid. So we have a presynaptic neuron and a postsynaptic well, really, it's really more like a postsynaptic cell, because it's not always a neuron. So the presynaptic neuron is the cell that is sending the information kind of towards the synapse, right? Before. The postsynaptic cell is then sending the information, receiving the information from the synapse. So that postsynaptic cell could be another neuron, or a muscle cell, like we saw with the neuromuscular junction, or even a glandular tissue, like, say, the thyroid or your sweat glands or something like that. So most neurons actually function as both presynaptic neurons and postsynaptic neurons. So most neurons are both sending information to one synapse while they are simultaneously receiving information from another synapse. There are literally 10+ billion neurons in your brain, so we've got a lot of connections there. And actually, one of the neat things that happens is as you learn, you actually make new synapses, which is pretty darn cool. So let's talk about the two types, electrical and chemical. In the electrical synapses, we don't have those neurotransmitters, we don't have a little synaptic cleft. We really more have a direct connection between the two cells, where they'll have some gap junctions between them, and so the information gets kind of flowed rapidly from one cell to the next. It can also be bidirectional, so it doesn't have to go pre to post, it can actually go post to pre. So electrical synapses are really, really helpful in kind of synchronizing activities, particularly when there's a lot of close contact between cells. So electrical synapses are much more common in embryonic nervous tissue than they are in kind of adult nervous tissue. So since at no point are we kind of messing around with neurotransmitters, the nerve impulse, it's all ion flow, so it's all electrical signals, hence electrical synapses. What we looked at when we looked at the neuromuscular junction was a chemical synapse. And so, of course, chemical synapses use neurotransmitter. In chemical synapses, it's a unilateral transmission of information from the presynaptic cell to the postsynaptic cell. Again, typically an axon terminal to, say, the dendrites of another neuron, or the motor end plate of a muscle, or the receptive part on glandular tissue. Because chemical synapses use neurotransmitters to kind of send the information across the kind of fluid-filled synaptic cleft, we have an electrical impulse in the presynaptic neuron, then this signal in the form of these chemical neurotransmitters in the synapse itself, and then that ends up causing depolarization, and another kind of electrical signal in the postsynaptic cell. So chemical synapses are slightly slower than electrical synapses, but I mean, we're talking on the order of like nanoseconds, not anything that you would actually like notice, at least without a few fancy equipment to measure it. So typically, neurotransmitters will affect their synapses for as long as they are in the area. And so to end the effect of the neurotransmitter, we either need to, we have to get rid of the neurotransmitter. So we either reuptake it, i.e. the axon terminal reabsorbs it. We degrade it using some enzymes, like acetylcholinesterase, as happens at the neuromuscular junction, or the neurotransmitter kind of just diffuses away. If you've ever heard of an SSRI, selective serotonin reuptake inhibitors, SSRIs are a very common class of drugs that are used to treat depression. What SSRIs do is they prevent the reuptake, selective serotonin reuptake inhibitor, of a neurotransmitter called serotonin. Serotonin is a neurotransmitter that basically makes you feel good. And so if you can prevent the reuptake of the serotonin and have it stay within the synapse, it can continue to have an effect and continue to make you feel better. So very, very common class of drugs to treat depressive disorders. Now, the kind of strength of the response caused by these neurotransmitters depends on how much neurotransmitter is released, and also, again, how much time it stays in the cleft. So if you only get a little bit of neurotransmitter, or it's swept away really quickly, or reabsorbed really quickly, you're going to get a small graded potential, potentially not even hitting our threshold value. More neurotransmitter, longer time in the synapse, you're going to get a bigger graded potential, much more likely to hit our threshold value, and then thus more likely to get an action potential in the postsynaptic cell. So there are two types of kind of possibilities on that postsynaptic cell. And you can see we have this particular neuron here. It's not getting just information from one other neuron. It's literally getting information from a ton of other neurons. And it's really the cumulative effects of all of these different messages that then results with what we call an EPSP or an IPSP, an excitatory postsynaptic potential or an inhibitory postsynaptic potential. The best situation to think of is like you've got like 5 people telling you to do this thing and 2 people telling you don't do this thing. What is the end result of what you're going to do? Because again, it's not just one neuron talking to one other neuron. It's hundreds and billions of neurons, tens of billions of neurons in your brain all talking to each other just about. So you might have guessed from their names which one makes things happen and which one makes things not happen. So excitatory postsynaptic potentials are essentially the fire, right? Like let's do this. So EPSPs bring our neuron closer to our action potential threshold. They make our neurons more positive, less negative, usually due to the influx of some positively charged cation like sodium. And so in this instance, we're getting closer to our threshold, so we have an action potential that's more likely to happen. An inhibitory postsynaptic potential is kind of the don't fire message, so don't do it. It drives the neuron away from the threshold value, more negative, makes an action potential less likely to happen, usually due to either the outward flow of a positively charged ion or the inward flow of a negatively charged ion like chlorine. But whether positive ions flow out or negative ions flow in, the end result is we're getting more negative, further away from threshold, and less likely to fire an action potential. So I'm going to go back here for a second. All right, so I want you to look at this image again. And remember, this neuron here, our postsynaptic cell, is not getting just one message. It's getting lots and lots and lots of messages. So typically, a single excitatory potential is not usually enough to get us over our threshold value to actually cause an action potential. However, all of these different electrical potentials, these graded potentials, can summate. They can add together. And so what we're really doing is we're getting kind of the sum of all the excitatory potentials plus all the inhibitory potentials. And if we get to our threshold value, then of course we will have an action potential. But if we don't have our threshold being reached, then we won't get an action potential. Let's say you had more inhibitory signals than excitatory signals. That would result in an action potential not happening. So there are two ways that potentials can summate or add together. That's spatial summation and temporal summation. So let's look at kind of each example here. So here, right, here is our presynaptic cell talking to our postsynaptic cell. It's sending an EPSP, but they're not really close together. They're not super strong in terms of their graded potentials. We don't ever get a threshold value, and so we don't get an action potential. Right? So, you know, you're getting fire, fire, fire. That's not really enough to get us going, as it were. In temporal summation, we get kind of rapid fire, or think, you know, more kind of submachine gun type fire. In this instance, we've got EPSPs that are close together, and we get wave summation. It's the same thing that happens with our muscles, right? So we get an excitatory signal, and it starts to repolarize, but before we can repolarize completely, we get another excitatory signal. That allows us to hit our threshold value, and once we hit our threshold value, then we will get an action potential. Now, EPSPs and IPSPs can add together spatially. Let's say you got a go signal at the same time as you got a don't go signal. Again, you'd have to have that summation to the end result, the net has to get us to our threshold value. In spatial summation, we're talking about more than one presynaptic cell sending excitatory signals to the postsynaptic cell at the same time. And so we're not just getting one go signal, we're getting multiple go signals all at the same time. That allows us to hit our threshold value. And once we hit our threshold value, we get an action potential. In this instance here, we have spatial summation of an excitatory signal and an inhibitory synaptic potential. So the excitatory signal brings us closer to threshold, but then we have an inhibitory signal that gets us further away from threshold, and essentially they kind of just cancel each other out, and nothing happens because we don't hit our threshold value. If we don't hit threshold, no action potential. So there are multiple ways that both excitatory signals and inhibitory signals can kind of summate together. So neurotransmitters are how our neurons talk to each other. Because again, chemical synapses are by and large what we use in the body. There have been over 50 different types of neurotransmitters that have been identified in the human brain. You can take a neurology class on nothing but neurotransmitters. Most neurons will make more than one. That allows them to have kind of several influences, right? If they can make both, acetylcholine and dopamine, then they can kind of do more than one thing. Usually it relates to the stimulation frequency. If you get one frequency of stimulation, the neuron will release one type of neurotransmitter. If you get a different stimulation frequency, the neuron will release a different neurotransmitter. So I mentioned serotonin earlier. Dopamine is another one. The serotonin and dopamine are both kind of the neurotransmitters that make you feel good. So when you're feeling happy and, satisfied, basically your brain is awash in serotonin and dopamine. So technically they're the only two things that you enjoy, because that's what's actually giving you that pleasure sensation. We typically classify neurotransmitters based on their chemical structure or based on their function. Neurotransmitters come in every conceivable format that you could possibly imagine. Some of them are proteins, some of them are just amino acids, so it's kind of parts of proteins. Some of them are gases, like nitric oxide. That's the kind of laughing gas if you go to the dentist. Some of them are purines, like ATP. Some of them are lipids. Some of them are amines. They really pretty much just have kind of every chemical structure you could possibly imagine. Some of them function very similarly to THC, the substance in marijuana that gets you high. So there's really no kind of limit in terms of their chemical structure. In terms of their function, we typically look at their effect or their action. So when we look at effects, are they always depolarizing? Are they always hyperpolarizing? Are they receptor dependent? So excitatory neurotransmitters always cause depolarization, make an action potential more likely to happen. Inhibitory neurotransmitters are hyperpolarizing, make an action potential less likely to happen. Some neurotransmitters are one or the other exclusively, but many neurotransmitters are dependent not on the neuron that releases them, but on the receptor that they bind to on the postsynaptic cell. So for instance, acetylcholine, when it binds to a muscle cell, is always excitatory. But if it were to bind to a cardiac muscle cell as opposed to a skeletal muscle cell, it can be inhibitory. So it just kind of depends on the receptor. In terms of actions, we look at direct action neurotransmitters and indirect action neurotransmitters. The direct action neurotransmitters open ion channels, so they directly change the potential of the neuron. So these are very, very rapid responses. Acetylcholine is one of them. Most of our amino acid-based hormones like serotonin and dopamine are like that. Indirect action neurotransmitters don't directly, hence indirect, affect the membrane potential, but they act through our second messenger system. So they take a little longer to kick on. Their effects tend to be kind of more broad and long-lasting, so somewhat more similar to hormones. Some of our dissolved gases work like that. So let's look at two examples. So here you have a direct action neurotransmitter. The neurotransmitter binds onto the ion channel, the ion opens, and we get an inflow of ions, and we get a depolarization or a hyperpolarization. In this instance, it would be a depolarization probably. That's what acetylcholine is, direct action neurotransmitter. We open an ion channel, directly change the potential of the neuron. In the indirect action neurotransmitters, the end result could be an open ion channel and a change in membrane potential, but it could also be going into the nucleus and activating the synthesis of new proteins and completely changing kind of the function of the cell. So in this instance, the neurotransmitter is the first messenger. It binds onto this G receptor linked protein. This G protein then goes and activates another enzyme that's embedded in the plasma membrane, which then releases something that we call a second messenger. Cyclic AMP happens to be one of them. There are several. And then that cyclic AMP can go open ion channels, it can go activate enzymes, it can go turn on protein synthesis inside the nucleus. And so we get much kind of more broad, kind of almost more systemic effects with the indirect action neurotransmitters. This process is a little bit longer. It's kind of like a relay race as opposed to a sprint, like the ion direct action neurotransmitters would be. So it's just a little slower in terms of response than the direct action neurotransmitters. So neural integration refers to the fact that neurons function in groups. Again, I repeat, 10 billion plus neurons in your brain. If they were all functioning on your own, you'd have basically a cacophony in your head and not be able to get anything done. But our neurons cooperate with each other. They work in groups, and that's part of what gives us some of these really, especially high-level thought that we have as humans. We really need this kind of integration for the brain to operate smoothly as a whole unit. And not just 8 million disparate parts, all kind of doing their own thing. So we call a group of neurons a neuronal pool. They get the info, they make sense of it, send it onward. A very simple neuronal pool would have a single presynaptic cell and maybe a couple postsynaptic cells. You can have much more complicated neuronal pools. So the neurons that are in the discharge zone are the ones that are really close to the presynaptic kind of incoming neuron, and they're more likely to generate the impulses, more neurotransmitter release. The facilitated zone are neurons that are further away and not usually getting as much neurotransmitter release, and are not typically excited to our threshold value unless they get stimulation from some other source. So here you can see a very simple neuronal pool with one presynaptic neuron talking to these several postsynaptic neurons. These three here are in the discharge zone. These two here and these two here are in the facilitated zones. They're a little further away. They may not be getting as much neurotransmitter release. And so these neurons are much less likely to fire an action potential than these three neurons here in the discharge zone. There are kind of two ways that neural pools process information. We have what we call serial processing, and we have what we call parallel processing. So in serial processing, think domino rally, basically, right? One input traveling along one pathway to one specific destination. One neuron stimulates the next neuron, which stimulates the next neuron, which stimulates the next neuron, right? The domino rally. This is how our reflexes work. We get kind of an all or none kind of specific, kind of anticipated response, kind of automatic, like our reflexes, right? Knock the patellar tendon with the reflex hammer and you kick your doctor. A particular stimulus, like that one, will always cause the same response. Conversely, we also have kind of parallel processing where we get kind of input traveling along several pathways. But let's look at this kind of reflex arc, for instance. So this would be a kind of serial type of processing, where the receptor talks to the sensory neuron, which talks to this integration center in the spinal cord, which then talks to a motor neuron, which then talks to some sort of effector organ. One to the next, to the next, to the next, to the next. That's serial processing. Everything's in a series. In parallel processing, we get kind of several simultaneous pathways. So in this instance, a single stimulus is not going to result in just one type of response, but actually multiple types of responses. Parallel processing is super important for all of our high-level thought. Let me give you just one example, right? Think of like your favorite scent, right? When you smell your favorite scent, not only are you getting all of the sensory information from the scent itself, maybe you have an associated memory with that smell. And maybe with that sense of smell, you also have an associated feeling with that particular odor. And so the stimulus, the odor, is not resulting in just one response, but in multiple responses, all the sensory data, the emotional data, the memory data, and you may even have a physical response to it, right? And so that requires parallel processing, much more complex than kind of that one to the next serial processing that we see in our reflex arcs. So there are 4 types of circuits, the connections in the neuronal pool that we typically find in a human brain. We have diverging circuits, converging circuits, reverberating circuits, and what we call parallel after discharge circuits. They kind of tell you in their name what they look like. So in a diverging circuit, you have a single input, lots of outputs. Very amplifying type of response. Really good example is your muscle responses, actually. A single motor neuron in your brain can activate several different motor neurons in the spinal cord, which can then activate potentially thousands of skeletal muscle fibers in a particular muscle, especially some of those really big muscles like your quadriceps or, say, latissimus dorsi in the back. So single input, many outputs. That's a diverging circuit. The converging circuit is the exact opposite. Here we have multiple inputs with a single output. So this is a concentrating type of circuit. We'll go back to smell. Right. So maybe when you smell, wood smoke and pine trees, right? Those are 2 very distinct odors, but both of them can result in, oh, hey, I remember camping with my family when I was 10, right? That's a converging type of circuit. Reverberating circuits, well, they reverberate, meaning they go through a chain of neurons to signal, and then it feeds back to previous neurons, so it oscillates. It reverberates like an echo. So the signal travels from this neuron to this neuron to this neuron to this neuron, which then also talks to this neuron, which then also talks to this neuron, and it's this kind of oscillating, reverberating, echoing pattern. All of our rhythmic activity, things like breathing, waking and sleeping, even walking, which is this very repetitive activity, is controlled by reverberating circuits. And then the last one is what we call parallel after discharge circuits. These are the circuits that are involved in our really high level thought, all of our doing math problems and learning about our brain. So here we have kind of a single input and kind of a single output, but we travel kind of multiple paths to get there, and so that's the kind of the parallel part, and the signal arrives at the output neuron at different times. So we get kind of a burst of signals. You get one signal and the next signal, and next signal, and next signal, so you get kind of burst of information, hence parallel after discharge. So that discharge refers to that kind of burst of signals, because all of the information arrives at the output neuron at different times. That is the end of chapter 11.